All right. Well, thank you very much again. Yeah, I think this is a great forum. Um, has many good resources that I'm planning to go back and look at some of those talks. But yeah, I'm happy to be able to share some of the stuff I've been working on with you guys. So just a brief summary of what I'm gonna be talking about. So I give you an overview of the presentation and then the R code. So in the presentation, I'm just gonna try to show you the different tools that I have been developing with our people for acoustic data analysis and trying to organize that across, well, along the what I consider the common acoustic workflow that we do for this kind of research. So I go over some of the packages and tools, and I have a few lines of code here in the presentation, but don't worry too much about, going on what is happening with that code, because then we're going to look at those, the packages, the code more in detail. So those codes are here just to make a few points, but it's mostly some broad explanation of how to organize these tools in a sequence that can get you from the recordings to data you can analyze statistically. So yeah, and I wanted to start with some basic questions that we share in this kind of research. So I think we all, well, there is a diversity of questions that we try to tackle with bioacoustic tools, and they can go from trying to figure out the community using passive acoustic monitoring in certain habitats to more like sociological questions about the factors shaping the evolution of signals and specific features, for instance, complexity, that has been a big topic for a while. But there are things more related to the ecological interplay of signals and the environment, and how those environments can shape the signals through their transmission properties. And of course, we also wanted, well, the topic that has been out there for a good amount of time is trying to understand how signals vary in space and time. We're talking about dialects or cultural evolution, and trying to understand also which factors are affecting those things. So this is not an exhaustive list of research questions that we can have or we can tackle with bioacoustics, but it's just to show you a diversity of topics that in which we can apply that. So it's quite diverse, but somehow I think we can narrow down the analysis workflow that we do for this kind of research like this. So I have very summarized version of these workflows. And for me, it takes these 5 steps, more or less. There is variation, of course, about how we do this, but we basically, first, we want to get the recordings, right? We need to get registered the vocal behavior or acoustic behavior of animals. That can be done also with synthetic sounds. For instance, for these signal degradation experiments, we can use synthetic sounds, or we can also get them nowadays from repositories, freely available recordings on repositories, and that's actually a big source of data nowadays for instance, comparative analysis. So we get the recordings, and then we need to to identify the position of the signals in those recordings, right? We have some target signals that are related to our research questions, and we need to be able to tell where they are to later tell the computer, go here and try to measure something, right? So those annotations are basically coordinates in time and in frequency. If you guys are familiar with spectrograms, these visualizations of sound in frequency and time in frequency. So those coordinates are a necessary step most of the time, except for some AI methods, but you need to tell somehow where those signals are, and that annotation step, although might be seen as not that relevant, it actually can take a lot of time to do, and it's changing a little bit in the way that we do it, thanks to deep learning on other tools. but it's something that can take us a lot of time. And once we do that, then we tend to measure things on those signals, right? Like, or sounds. I don't know if you like the word signal, but I'm using signal in a very general way. So we measure things on them. We need to quantify the structure in most cases, either because we have a specific hypothesis or predictions about how that structures vary between groups or individuals, etc., or because we want to use those features for doing, for instance, automatic species detection. But we need to measure something, do some quantification, and sometimes that quantification requires some additional transformation of data that I'm including here as a independent or as its own step, kind of isolated from struct features, because sometimes we have so many features and we have multidimensional traits, or the sounds are represented as multidimensional traits, and sometimes we need to do some dimensionality reduction to be able to adequately process the data and being able to run stats that can deal with more simple representations of the traits. For instance, latent spaces, right? Like PCAs on a bunch of acoustic parameters or features. And once we do that, we can, in most cases, run stats or descriptive analysis or whatever. But for me, this is kind of the most common workflow. And I'm pointing out to this workflow because that's how we're going to be presenting some of the tools that I'm going to be talking about. And for me, that's how it makes more sense when to talk about specific tools, in which step you can use those tools, right? Same thing when you're talking about tools to fix your car, or which specific context you can use those tools. So what I'm showing here, what I just added to this diagram, is the name of some of the packages that we have developed. And you don't have to remember this, but basically we have packages for many of the steps for this analysis workflow of bioacoustic data. We can use Warbler and Baruli, in fact, to synthesize recordings. You can help the annotation either through automatic detection with a HUN or importing things from Raven with our Raven. And these are packages like Warbler, Baruli, and Phenotype Space can help you do this feature extraction and further quantification of latent spaces. But I'm going to be talking about this for the rest of this talk, so don't worry too much about this. Is too much information, I know, but that's the way that I'm organizing this stuff. There's another package, Dynaspec, that you can use for creating dynamic spectrograms, basically videos. But that's mostly for presentations. It's just to show some fancy spectrograms, but it doesn't add that much to the analysis. An important point here is that these first two steps are usually the most painful for many people, including myself. And because it takes, getting the recordings is going to the field is never easy thing, but also the annotation step is not always straightforward. For many causes, for instance, sometimes we want to try automatic detection, and in some cases it works, and in some cases it doesn't. Actually, I think we're in a good time because many people is developing different methods to do automatic detection, but it's also hard to navigate those things. And I'm going to be talking about the method that I have developed, but there are some other methods and some way more powerful. But it also has some trade-off about more powerful, but they need more data, and you need to find the right method for your data, and that's not always straightforward. And manual annotation is always like the baseline way to do these things, but it can take a lot of time. So choosing the method and how to do the annotation is also is something that can take quite an amount of time. So that's what I'm highlighting those two, because for me, once you're done with that, the rest of the steps are pretty straightforward. Of course, you need to figure out which features you want to extract, but basically running the code for doing those sort of steps is relatively easy compared to the annotation steps and getting the recordings, the previous steps. So that's important to keep in mind. That's something that I'm going to be emphasizing. But it's also something good that once you have those annotations, things shouldn't be as painful. Okay, so first I'm going to talk about some ways in which we can do annotations in some files. I'm pretty sure you guys are familiar with annotating. Well, people have been working with the bioacoustic data has passed through some sort of data format like the one that I'm showing here. So this is the basic annotation format that we required for getting those coordinates of the positions of sounds in these recordings. And the basic format is something that this is the thing that we're gonna fit the computer with, and is the way that we're gonna tell the computer go look at these specific positions, right? So that's it. And the computer, to be able to do that, it needs to know what sound file we're talking about and where the sound starts and ends in that sound file. So that's kind of the basic 3 pieces of information. And because we usually have more than one annotation per sound file, we want to add a unique ID for each of those annotations. So that's it, like 4 columns, start, end, and sound files, and unique ID for each one. And it looks simple, so it's kind of weird that I said that it's hard to get here, but yeah, depending on the amount of data and the variation of your signals, it can still be difficult. So that's the basic format of an annotation table, but of course we can add some additional information. For instance, the channel, if you have stereo recordings or multi-channel recordings, you want to specify on which of those channels the sound is found. And something that is common is to include information about the frequency range of the sounds, right? If you can focus your analysis on a specific frequency range, that probably is going to provide you more accurate measurements on the structure of those signals. So those are the top bottom frequency columns that I'm showing here at the end of that table. So that's the basic format, the basic information that we want to get from about the sounds that we want to analyze on our recordings. And there are several ways in which you can do that. So this is just the 2 that I'm going to be talking about. There are many packages, mostly in Python, to do automatic annotations. So I'm just measuring here the methods that I tend to use and the ones that are related to the packages that I'm going to be talking about. But that doesn't mean that this is an exhaustive list at all. So the 2 options that you can easily do through R is first doing a manual annotation through Raven. Raven is a software with a graphical user interface in which you can do boxes like this image that I'm showing you here. You can do boxes around the signals in the spectrograms and tailor those things in the waveforms. For those that are not familiar with the spectrograms, that's the view that you have in the bottom panel. And yeah, it's showing you the variation in frequency through time. So it's a graphical representation of sound, and the one at the top is the waveform, which shows mostly the changes in energy through time. But Raven is a great tool, and it has a light version that is free for everyone that allows you to do the annotations. So that's accessible. And the good thing is that we have this R package called RRaven that can import the data that you annotate in Raven. Those are called selection table files or selection files that come in .txt format. And with this package, RRaven, you can read many of those selection files at once and import that data into R pretty easily. So that's one kind of workflow that I use all the time when I think I have to do manual annotations, or when I'm like tailoring some automatic annotations that were not that great. So some mixture of automatic annotation and manual annotations. And this is some of those codes that I said that I'm gonna show here. We're gonna try this in the R code that we're gonna go through later. But this is just to show you with one call of RRaven. If you just point out to the folder when you have all the selection files, then you can have all that data imported into R. And you can even do it in a format. I just added, I don't know if you noticed this, but into the call of imp Raven, I added this argument, Warbler format, and then the name of the columns change to add or to use the format that is Warbler friendly, or the column means that Warbler is expecting. And it's also changing a bit some units. For instance, Warbler expects kilohertz and Raven uses hertz. So just a few adjustments so that data is ready to be analyzed with Warbler, for instance. So that's what I usually do that, like annotating Raven or fixing things in Raven and then going to R with RRaven, and then I can use that with our packages for data analysis. Something else I wanted to point out here, I'm gonna be using this kind of syntax. I don't know if you guys are familiar with this. You guys know this is a function, right? This is an R function. But when we include this with double semicolon, that means that this function comes from this package. This is not something you have to do all the time, but I'm including that so you know each function, any function that I include here, you can tell from which package it comes from. So I think it's a bit more informative. All right, so with Raven plus RRaven, I think it's a good, it's a good combination that can help you can allow you to use Raven for all the annotations and then go with the rest of the analysis into R. It also allows you to go back to, export things in a format that can be used by Raven, so you can do things in R and then go back to Raven. I will show you some code about that. So it's not only in one direction. You can go from Raven to R and from R to Raven as many times as you want to take advantage of those two tools. Okay, so if you decide to do some automatic detection, I'm going to be talking about OHUM. And again, there are many other tools to do this kind of stuff. But the one that I'm showing here, I just developed this recently for some analysis. I'm working with frogs and passive acoustic monitoring. Despite having some relatively traditional or methods that have been out there for a while, like template-based detection and energy-based detection, it can reach some decent performance. And that is done based on trying to optimize the detections using reference annotations. So the idea is that you have a big set of data and you can take a subset and annotate that subset, annotate all the sounds in that subset of data. With that data, I mean a bunch of recordings. And then you can try different detection parameters on that subset of data that has been annotated and figure out which combination of detection parameter values can provide the best detection, right? And because with the reference data, you know what should be detected, right? So that's the main point of the package. And for doing that, it uses signal detection theory. It uses this true positives, false negatives, and some indices that are derived from those, like recall, precision, F1 score. And with that, you can get a sense. You try a bunch of different parameters and try all possible combinations of those parameters. We will run something to show you how that works. But then you can pinpoint the combination of parameters that gives you the best performance. And then you can apply that into the larger data set. So that's the basic idea of logic or rationale of the package. And of course, it needs first to do some annotation of this training data set, a subset of the whole set of recordings. And then you will compare those references to the detection. So that's basically what I'm showing here, right? Like here's a detection that got out of 10, it got 7 detections. And you can iterate that. For this specific sample, it means that it has a recall of 0.7 and good precision. Everything that it got was the right type of signals. These are basic signal detection theory indices, so we don't have a lot of time to go over these ones, but for anything related to detection of species or things like that, you probably seen similar indices. And then, yeah, so what I'm showing you here is some measurement of the performance of the detection. And what I said is that you can do that, you can iterate that with different parameters to figure out which ones will give you the best detection, right? Like the one that is closest to the training annotation data. And it's relatively easy to run. So here I'm showing you an example of a template-based detection. So template-based detection uses cross-correlation, which is taking the spectrograms, could be mel frequency spectrograms or just Fourier spectrograms. And it takes a template, an example that you think is similar to most of our other signals. And then that thing is run over the recordings. So it's doing a comparison of the amplitude values for that template compared to each of the steps across the recording. And that generates in this figure that I have down here, the lines below the spectrograms, that's kind of the peaks that you will expect when you find some good match between the template and a sound in the recording. So once you do that, then you just have to come up with a threshold that you decide that everything that goes above that point is going to be identified as detection, and below is going to be ignored, right? So that's the basic logic for this template-based detection. We're going to run an example again, so don't worry too much about that. But what I want to show you is how you do this optimization. So in this case, I'm running the code in which I said that I want to try different thresholds, right? I want to try 5 thresholds going from 0.1 to 0.5. Those thresholds go from 0 to 1, and 1 is the highest, right? So you want to try something between 0 and 0.99. And once you do that with this function, optimized template detector, it will provide you, well, the output is going to be something like this, in which you have for each of those thresholds, these diagnostic indices that tells you which of the parameters, well, which of the threshold had a better performance, the best performance for detecting those signals based on the reference annotations that you provided. Okay, so in this case, we can see that the higher thresholds, 0.4 and 0.5, they give you a better performance. It is detecting everything that is detected is the right signal. So F1 score, which is the combination of these two other indices, of 1. Yeah, so that's the way that you can do this kind of annotation. Remember, we're still talking about how to get those coordinates of the signals in the soundfast. So if you want to go, you're going to try some automatic detection, you can do it with a method like this, with a home, with this package. you need just to try different parameters to see what's the better approach, or if you can actually get a good detection performance for your specific data and templates that you're using. Okay, so this is an automatic detection method, but I think it's also good even if you want to, or you want to try a more powerful approach, for instance, with deep learning, many tools that are being developed in Python. I think it's a good starting point to get used to how these things work and to understand how training data can be produced and used. Actually, same training data can be used for this method or for other methods. And but the good thing is that it tends to require less data than other methods that are more data hungry, right? With AI, you tend to need way more samples, more and more examples. Although there are some new methods for transfer learning that you can have pre-trained methods, but that's not that easily available, I think, in bioacoustics. Hopefully, there's going to be it's going to be moving faster, and we will have more powerful methods that are available. But again, this is a good first step, and for many cases, it could be good enough to do your detections. And this is this is the last step that I will do for this example that I show you here. So I'm playing with different thresholds here, and I figure out which threshold is good or for this detection. And once I have that threshold, I can apply it to the whole training data set. And of course, once I feel comfortable with that, the way that I train that method, I can go ahead and run it on the complete data set. And in that way, you can analyze these big data sets that we tend to have, for instance, for passive acoustic monitoring. So that's the second option that I'm showing you here for acoustic detection. Again, we will go through some code on how to do those things. So I'm just showing you an overview of these tools, so you have it in mind once we start doing that. And as I said, that's kind of the first steps, trying to figure out where the signals are. And once you have those coordinates, time and frequency, and in which sound files, basically annotation tables that are referring to the signals we want to analyze, then we can go ahead and analyze in some way those signals that we call feature extraction. But also, I'm going to be talking about other methods to quantify variation related to latent spaces that I mentioned at the beginning. So for that, I have two packages, Warbler R, Barulho. Well, basically, I think the package that people is more familiar with is Warbler. I'm missing an R there, but Warbler is a package that makes use of those annotation tables to measure different features of the signals that can be later used to represent latent spaces or basically do our stats. And the way that it does it is basically iterating tasks with a loop over each of the rows of those annotation tables. So you guys probably run loops in R, so it's the same idea. You have rows that indicate that if you go to this sound file and to this start and end time and this frequency range, you find the signal that I'm caring about. So go and measure something. In this example, we have signal to noise ratio, SNR. But most functions will work like that. And that's what I said, once you get to the annotation tables, things tend to be easier because the functions are developed to work on those type of objects, data frames or selection tables. There's a specific class of object, but they all have the same information, and you can apply different functions to extract different features using the exact same data format. And it not only do this feature extraction, but it also both Warbler R and Barulio, that is another package that's for quantifying degradation of signals through transmission. But they both take these annotation tables and they do the measurement of those tables using this iteration of tasks with loops. And they can also produce spectrograms or visualizations of image files that can allow you to double check that the things you are measuring kind of make sense. And there are some parameters or some features that we extract that can be visualized on spectrograms that we will see when we run the code. So I think that's also a special feature of these packages, that is not only measuring things and trusting that things are measured correctly, but there are ways in which you can double check things. You can make sure that you are measuring more or less the right thing. So that, to me, that's an important step. And this is just an example of what I was telling you. Once you have annotation table, then with a single line of code, you can get some features. And this is the detections that the file, well, our object that we created with Ohum, this automatic detection that I had the example before. So I can just input that into this Warbler function, which is called spectral analysis, and it will extract a bunch of features. Here I'm not showing all the columns, but it extracts some 27 features related to the distribution of energy in the time domain, in the frequency domain, and also to the frequency contours like dominant frequency, fundamental frequency, and a little bit, if you want, that's an additional feature that you need to call with a different argument. But you can also measure features of the harmonic stack, the harmonicity of the signals. Yeah, but that's something you would do with Warbler, and it can take only a single line of code once you have those annotations. Again, I'm emphasizing this thing that having the annotations is the hardest part. Once you are there, things should be easier. This is another example. Same thing with Warbler. You can measure this spectrographic cross-correlation, which is a pairwise similarity metric. So it tells you, the previous one is kind of an absolute measurement of the structure, like what is the pitch, what is the duration, things like that. But this cross-correlation tells you something more like the pairwise similarity. It only makes sense as a comparison between sounds. But it basically tells you how similar two things are based on their spectrogram. And that can be a very powerful way to quantify structures. Mostly when things that can be represented as a single sound, like, like single sound repertoires or things like that. In this case, it gives you a dissimilarity matrix. A similarity matrix, sorry, in which the diagonal means things that are very similar. Well, one means things that are the same. So the diagonal is that because it is the comparison to itself. So higher values tells you things are more similar, things that are closer to zero are more different. So there are a couple other functions to do feature extraction that we will look at when we run the code. But again, it's just to show you that measuring things can be easy once you have those annotations. And you can do those things with Warbler. For some features, with Barulio, you can do other features, but those are more specific for these signal transmission experiments that unfortunately we won't have much time to talk about that here. Okay, so feature extraction is also something you can do at a different level. If you have many features, sometimes it's hard to deal with those things with more traditional statistics, or the statistical tools that we have for multidimensional traits are not as flexible or powerful, right? Like mixed models and correlation structure, things like that are not easily applied when you have these multidimensional traits. So sometimes we want to reduce dimensions and even quantify those multidimensional spaces in a way that you can end up with a few measurements that are easier to be used in those traditional statistical methods. And one way to do that is representing these multidimensional spaces or multidimensional traits as latent spaces. This is an example data set. Actually, this is simulated, so this is too good to be true. We never have such a good rounded groups that homogeneously spread in the the acoustic space. But it can help me make the point here. So if you can have different groups, what I'm showing here with the G's, G1, G2, G3, you can quantify several aspects of the acoustic space they occupy. And acoustic space, I'm going back and forth between latent space and acoustic space, but it's just the trait space in a more general sense. For instance, you can quantify how big are the spaces they're occupied. That can tell you something about the diversity of the sounds that are found in those groups. But you can also measure somehow the similarity of the different spaces. For instance, that can be done by measuring how much they overlap, or what is the mean distance between points from different groups, or distances between centroids. There are different ways in which you can do that. But then you end up with a smaller or lower, a data set with lower dimensionality that might be easier to handle with more flexible statistical approaches. So that's the point of this sort of package phenotype space, trying to measure those additional features of the acoustic spaces of traits that are multidimensional. Then you can go ahead and project those things in a few dimensions, and then measure somehow the similarity of the spaces that are occupied. And one example that I guess you guys are probably familiar with is when we have many features, many parameters, and we end up doing a principal component analysis, or we do like a multidimensional scaling analysis in which we end up with a few vectors, like that could be the dimension 1 and 2 that we have in this graph, right? So that's how the goal of the package, things that can be summarized in a few dimensions, then you can extract measurements or that characterize their space and their similarity between different groups. And this is just one code just to show you how that works. This is a function from phenotype space that tells you the space similarity. And there are different methods to measure the similarity. This is the density overlap, which is using kernel densities to measure the overlap between acoustic spaces, which takes into account the density, right? Not just the area in which they are found, but how dense are different areas of those areas, or the regions of those areas. And this is what you end up, the kind of data you get with this function. So it tells you for each pair of groups, the overlaps. It depends on which measurement of similarity you're going to use. There are several measurements, but for this one, it tells you how much the first group overlaps to the second group, and the other way around in this column. And this can be converted into a triangular matrix with this function, rectangular to triangular, but it's basically the same idea, right? But this is telling you showing you in a triangular kind of dissimilarity matrix, which how much overlap you have between the different groups. So again, I'm going not explaining too much about this code because we're gonna run it very soon. But the point here is that once you measure a bunch of features in the acoustic signals, you might end up with this huge or highly dimensional data set. But you can still do some measurements of characterize those spaces or the similarity between different groups, individuals, etc. And you can do that with phenotype space. Of course, this is not something that applies only to acoustic signals. So any kind of multidimensional trait can be summarized in that way. But I think I'm showing this here as part of this workflow that we tend to, many cases, we usually end up with many features, and we somehow need to reduce those dimensions and measure things on those new spaces. I'm gonna move to the R code, but I just wanted to point out two other tools that we have for acoustic analysis. Again, I'm not being fair here. There are many other tools that you can use. For instance, well, the packages that I'm using, they rely heavily upon other packages like Tuner and C-Wave. So those sort of packages have tools for doing small measurements or modifications of sounds that I put together, that I use as building blocks for my packages, but I need to be acknowledged. And they're pretty cool. So if you want to move into doing your own code or your own analysis, I definitely recommend you getting familiar with these packages, Tuner and C-Wave mostly. Then you have bioacoustics that it can allow you to do other measurements. And Monitor is another package for doing automatic detection. So that's definitely something you want to check if you want to explore other tools for acoustic analysis in R. And in Python, well, you have many things, but I just listed a couple of things here that are somehow similar to what I'm showing you here. Like I know scikit-mat can do this kind of feature extraction, and Biosound do a similar thing. So those are two packages that it will be worth exploring if you want to get more into analysis in Python, acoustic analysis in Python. And before I moved into the R code, these packages have been developed as collaborations in many cases, and so I want to acknowledge all those collaborators that have helped me a lot, or we have collaborated to be able to create these packages. Okay, so I'm gonna move to the into the code now. So I think this was already said, but the code is found in the GitHub repository for the seminars, right? So you can download it from there. There is a folder with my name, and then you will find an Markdown file, which is this one that I'm showing. Well, actually, it's this one here. And there's also a data folder inside that folder, that if you want to run this, you need to download that data folder and put it in the same directory in which you're going to have the Markdown, because that's the way that the function, well, the code is written. It's going to be looking for files. It has, the data folder has many example files, so it's going to be looking for those files inside that folder. So I'm installing, well, I already did this, but if you want to run it, I recommend using the latest developmental version, mostly for these two that I recently updated. And this code here is just defining the working directory. And that's what I was telling you, that we're going to tell R that it has to look for a subfolder from the current working directory for these example files, and also for saving some of the files that we're going to create. Okay, so the code here, well, we're going to start showing you how to do this importing of Raven files using our Raven. So in this working directory, that data, well, the data folder, you have these four selection table text files that are in the format that Raven uses for for the output, for outputting those annotations. Okay. So the way that this function works in Raven, so that's kind of the main function in our Raven, is basically you need to tell in which directory it has to look for, and then it will import every single file a selection table in TXT format, and is going to import all those selection tables in a single data frame, right? So, well, there's a way that you can have in as a list, but by default, it will include everything is in a single data frame, which I think is convenient instead of having to read each of the files once at a time, right? So with this line of code, you can import that data, and you have it in these object cells. As I mentioned before, there is an option to do to import those things in a format that is already that has that's friendly for using it with Warbler. So what we call here with this argument Warbler format. So the format you end up having here has slightly different column names. And also, as I said, it will format the frequency values. So it's in kilohertz instead of hertz. But that's basically what it does. It changes the columns and modifies some units. And this kind of data is ready to be analyzed with Warbler. So, yeah, it's pretty straightforward. It doesn't take too much to import those files from Raven. You can also use the argument files to refer to specific files. If you don't want to import everything that you have there, you can say, I just want to import these 3 files. And that's what I'm doing here. So I'm just have those data from 3 different files. I'm importing that into Warbler format. And that's pretty much it. That's the way that I'm use Raven most of the time. I'm going to show you how to export data, but just to show you that once you have this data in this format, then you can do analysis like this one. Here I'm measuring a bunch of acoustic features, like the duration, like the one that I show you here is the same, that I showed you before is the same function, the spectral analysis. So I'm showing you how to, once you have the data in the right format, using Raven, then you can just input that into the functions from Warbler and get features that are representing the structure of the signals, right? So here you have, let me see. I'm gonna save this in an object so we can look at it. And then I can do view and so that's the output of that function, and we have these many columns. It says there are 28 columns, so it's like 26 different acoustic parameters. And then as I said before, these parameters, well, there are many, so we cannot go one by one explain what they measure, but in general, they tell you things about the variation in energy in the time domain, in the frequency domain, and also about the frequency contours. With frequency contours, I mean like the dominant frequency and how it goes up and down through time. And also for, if you want, you can do it for fundamental frequency and harmonics. So, but basically a bunch of features that somehow quantify the structure of the signals. That's one of the methods that I talked about before. I also mentioned cross correlation. And again, with the same, this object that we got from imp-raven, we can run cross correlation and get these pairwise similarity measurements that tells you, which of those sounds look more similar, of those pairs of sounds look more similar to each other. Again, a pairwise measurement. So this is or triangular matrix, but there are many methods that you can use on that. For instance, mental test. Mental test allows you to compare this acoustic distance matrix, for instance, with geographic or spatial distance matrices to see if individuals that are closer together, they have more similar sounds, which is one of the predictions that you will have, for instance, when you're looking at dialects, some dialects. So that can be something that you can use already for testing some of your ideas. So that's cross correlation. Notice that I'm using this path WD. This is what we define up here. And I have the sound files there. Of course, these things are being measured on the sound files, right? Like every time that R goes, you provide these selection tables, R will look for the sound file, and you need to tell R where the sound files are located, right? So that can be a source of troubles at some points, but yeah, just make sure you are telling the right directory where to look for the sound files. And as I said, R-Raven I use it mostly for importing data, but you can use it also for exporting the data. So let's say that you did some measurements on these parameters, or you did some detection with Ahun. You didn't do the annotation with Raven, but you want to use Raven to double check the annotations or to modify slightly some of those boxes that you end up having for those detections. So you can use for that X-Raven, another function from R-Raven, to export the data in a Raven-friendly format that then you can open with Raven. So that's what I'm gonna do here. I'm exporting this cells data, telling the path where the sound files are located. Also, there is the path is where I want to save the text file. And you also need to tell the sound file path and the name of the file that you are exporting. And this is being exported as a multiple sound selection table, which means that is a text file that has data for many sound files, not for a single sound file. And you can open that into to Raven. So I'm just going to show you that quickly here. Raven sometimes gets slow. So this is the file that I just exported. Oh, I'm not seeing the name down there. Okay. And you will see that, well, I'm gonna use a different color palette so you can see the boxes. So this is kind of the default view in Raven. You have this selection table, which is the data that we have in R. And it's showing you the boxes here. I don't know if you can see this dotted green line, but that is showing you where the different files are being bind together. So you have several sound files. You can also see that, well, the font is pretty small, but you can see we have three different sound files here. So in that sense, you can export data from R to Raven. And for instance, if you have detection from Ahun, you could modify or just double check those selections here, or you could go here and say, all right, I want to play a little bit with this. I think the position is not the right one. So you could adjust things and then save it, and then go back to R, import that into R using in Raven, and so on. So I'm not saying that you have to go back and forth so many times, but the point here is that you can exploit R-Raven as a more flexible way to look at spectrum annotations, but then you can always go back to R to do the rest of the analysis. So, and that's the way that I intend to use Raven. And this is the example that I have here, that after you change things, then you can go back and import things into R. So in that thing, you can kind of integrate Raven as a graphical user interface for annotations, but still being based in R. Okay, so those that code was mostly for annotations, mostly showing you our Raven and Raven, how to go back and forth. So I mentioned this package. This is for automatic detection. So previously, if we end up doing manual annotations, but if we want to try some automatic detections, we can do that with this package. As I said, it uses reference tables. So you have annotations that then you can use to compare different detections and figure out which gives you the best performance, right? And once you have that, you can apply those parameters, detection parameters, to the whole data set. Here we are just loading some data examples that comes with the package. So these are two files that actually we already have those files in the example data, and the reference table. So these annotations for training that refer to sounds on those two files. There is a function that is called feature reference that gives you some summary of the annotations that you have in a reference table. Well, actually, you can do that with any selection table or annotation, but it's useful for trying to figure out parameters for that you're going to use to train the process, right? In some cases, I think I'm using, for energy detection, for instance, we need to tell the function that we want to keep only sounds that are within a specific duration range and a specific frequency range, and they have gaps of a specific duration. And sometimes, we can use our knowledge of the systems. You know that the species have, low pitch signals, then you can use that to narrow down the possibilities of values that you're going to use. But you can also use this function to get some sense of what are the features in the annotation or the reference data, the reference annotation. So this function tells you here, feature reference, that you have a specific duration of the selections with a mean of 163. This is in milliseconds. That's important to say because, we don't have signals of 163 seconds here. But it gives you some idea of the mean and the variation for different parameters that you can then use to inform the range of values that you're going to use for training, for optimizing the detection process. So it gives you that the mean, minimum, average, and maximum for the duration of the signals, for duration of gaps, the number of annotations that you have, the bottom top frequency, and so on. So that information is can allow you to get a better sense of which ranges you should be using for training, optimizing these detection processes. This function here is going to show the spectrogram of the, let's see, it's the same spectrogram that I showed you before, and it only works for short recording. So this function that I use mostly for the vignette of this package. So I don't think it's going to be useful for long recordings, but I wanted to show you here this because it's the same idea of annotating everything you have in the training data set. It wouldn't make sense to leave some aside, right? Because then the method is going to find those ones and you're going to think that it's wrong. So you need to be exhaustive about annotating any single target sound on those training data sets. So we have two files. I'm just printing the two spectrograms here, but I'm going to skip this second part. That's just the spectrogram of the second recording. And this is the actual line of code that do the detection optimization, right? So this is called optimize energy detector. So as I said at the beginning, there are two methods, using templates or using energy detection. And energy detection, the basic idea is that sounds that of interest are likely to be of higher intensity than the background noise. And those are the cases in which these kind of methods work, right? That when the signals of interest are louder than the background. If that's not the case, this is unlikely that it's going to work. But if that's the case, then you can use additional arguments to narrow down the search. So you want to pick up things that go above certain frequency threshold. This is given in percentage, right? So it's dividing the frequency range between 0 and 100%, and then you give some numbers to define which to which point you will determine something as a detection. And because this function is optimizing the process, is not testing a single parameter, it's testing several parameters for each argument, and actually is testing different combinations of those parameters. For instance, we are trying 3 parameters for threshold, and we are trying for smoothing. The smoothing is a way to take those amplitude values and make it more rounded. And we are trying different parameters for smoothing as well. So what the function will do is try all possible combinations of these thresholds, 3 thresholds and 3 smoothing parameters, values, to figure out which ones work the best. We also have a bandpass here, so it's going to be focusing between 1.5 and 9.6 kilohertz, and a single value for minimum duration. But you can have also several values here, and I recommend to go to the help for this function to see which parameters accept several values that you can optimize. But if you run this, so it's gonna go over all the files. Because we're not telling which files to work on, it's going to work over all the files that are found in the path. And it's going to give us at the end this signal detection theory indices that give us an idea of the performance of the detection. So if we look at this output, I don't know if this looks very small for you guys, the font. But this is showing us the different, for each row is going to give us the detection values that were used for a specific iteration. So this one, the first one, has a threshold of 2 and a smoothing of 5. And then we can go and look at how good was the detection. We can focus, it has several indices, like the basic ones about true positives, false negatives, and so on. But it has some other features like merge positives if they are, you have some detections that are merged or split. If you have one signal that has to be detected as a single one and is detected at more than one detection. But yeah, just for the sake of the example here, I'm just going to focus on these last 3. And you can actually go and look for the ones that have the best F1 score, which is kind of single metric of performance that you can use as a proxy to figure out which combination was the best. And then you can go and check those values for the parameters in which you play with different values and use those values for detecting on the complete data set. Basically what I showed you before, but just showing you a little more about the arguments and the way that this optimization works. But notice always you have to provide this reference, right? You need to tell the function, we're going to be optimizing based on this gold standard. This is the perfect detection. Tell me which of the detections get the closest to that one. That's basically what the function is doing. And the same thing is done with template detection. But we won't go over that now because of the time we have. This package Warbler that I'm talking about, that I talked about before for feature extraction, has some other features for getting, for playing and formatting and exploring our data sets, our acoustic data sets. I'm going back to this. Just to do some more smoother transition, we were talking about detections here, right? And we talk about annotations, manual annotations, or automatic detection, but at the end you will have a annotation table in which you have the coordinates in time and frequency of the signals you were looking for, right? So regardless if you do that manually or if you do it with automatic detection, you have that, and that can be input into Warbler in the way that I show you above. There are features that you can use from Warbler to format and explore a little bit your data. And this is what I'm showing you here in this chunk. For instance, Warbler options is a way to determine global parameters for all downstream Warbler functions. So you make sure that all analyses are being run with the same parameters. For instance, window length, which is a parameter that is used to produce the spectrograms. And if you change that, the measurements might not be as comparable across different sounds. So those are things that is important to keep constant. You can do it just with this call, Warbler options. You can say the overlap. The overlap is also another parameter for building those spectrograms. You can say 90%. And you make sure that all downstream analysis, Warbler analysis functions will use the same parameters and things will be comparable, right? You will be consistent throughout the analysis. There are functions like check sound files that tells you the sound files can be read in R and gives you some additional information. For instance, we have some files here that have a different sampling rate, and that can be problematic, for instance, for cross-correlation. So that is some, like sanity check steps that I think are can save you a lot of time, because some other functions might not be as informative about what is the error you are getting. So it's better to double check those things ahead of time. Info sound files can tell you things about the duration of the sound files, the sampling rate, the number of channels. So in this case, each row is one of the sound files that you have in that working directory. And it tells you for each of those files, these features about duration, sampling rate, number of channels, bits, the size of the files, number of samples. And for instance, we can pinpoint the file that seems to have a different sampling rate that was suggested by the previous function, which seems to be this last one here that has a 44.1 kilohertz sampling rate. And the rest, they all have 2205. Well, this is 2205 or 22.5. There are different sampling rates there. And you can do that easily with this. You can also consolidate. I won't run this, but if you happen to have many files in different folders, or day one, individual one, and day two, individual one, and things like that on different sites, Warbler is expecting all the sound files that you're going to analyze in the same folder. So you can use this function consolidate to tell the function, well, I have a bunch of files in many subfolders starting from this folder. So go and look for every sound file in all the subfolders and put that in a single folder. So you can consolidate the data set with this function, and you end up also with a CSV file that tells you where the files came from what was the original folder, so you don't miss that information, you don't lose that information, but it still can end up with all the files in a single place. And this is very simple, just to get the duration. So it's similar to the previous one. So you have these additional tools that you can use for to understand your data, acoustic data, and to format the acoustic data. And some functions for quantifying structure, feature extraction that we already did above, mostly for showing you how to go from annotations, how easy it can be to go from annotations imported into R with R-Raven to measurements. So that's what we show that up there. But here are a few more examples. So this is what we did before, the spectral analysis. So I told you about these 26 different features related to distribution of energy in time and frequency domains. You can also measure male frequency capsule coefficients. This is something developed for quantifying mostly the human voice, and it has been used a lot for voice recognition, but it seems to have important, or can be useful for quantifying animal acoustic signals as well. And we as a community are starting to explore those things, but it seems to be helpful, mostly on species identification and automatic detection, things like that, but also for other things that we do, for instance, in behavioral ecology and things like that. You can measure signal to noise ratio. You know how noisy is how good is the recording. Things that have high signal to noise ratio, you have lower background noise, so it's more likely you will have better quantification of the structure with higher signal to noise ratio. These are measurement dynamic time warping. Tells you it's another pairwise similarity metric. But that's based on the frequency contour. So if you imagine the signals as change of frequency through time, and you can measure the frequency at each step in time, you have these contours of the signals that then you can compare with a time series method that is called dynamic time warping, which tells you how similar are those time series. So that's what the function does. Mostly for things that have pure tones, right? Like they don't have a lot of harmonics. And other things that you can measure, simple things like the gaps, the distances in time between elements within the sound. I don't know how much time I have left. I have a couple more things, but they are not critical, so we can go over those or questions. That would be fun too.
Yeah, so Marcelo, we have like 7 minutes till we reach our time limit. So let me reload the page here. I don't think there are questions. So if anybody has questions here in the Zoom call, please post them on the pollev.com/EFI 2022. I can start with a quick question while we wait for to see. Have you ever run into problems where the files are too big and to bring into R, and if you have, what have you done, or do you have suggestions of how to deal with that?
Yeah, that's a great question. Yeah, I have had that problem. So for things on which you already have annotations on those files, it shouldn't be a big deal, because at least for the way that in which Warbler works, Barulho and Warbler, those two packages that measure things on an annotation or selection tables, because it will go and just read that specific segment, regardless of whether you have a 3-hour recording or just 2 minutes. If you have like 1 second sound, it's just gonna kind of clip that thing and measure it and then goes for the next one. But for the automatic detection, it can be problematic. So you have to be careful about that. And there is a function that can help you deal with those things. Let me see if we can go back to, yeah. So there is a function, let me see, that is called split acoustic data. And with that, you can tell the working directory. And if you have annotations, segments, okay, yeah, if you have annotations, I wasn't sure about that, but it's true. You can include that here, like you say, you have annotation tables related to the original long files. It will split the long files. For instance, you can tell the function to split that in 60-second segments. Or you could also use this argument not to split it in specific, durations, but split each one in 10 different segments. and it will split the sound files. It will create a new folder with those clips, and it will also give you the annotation data adjusted for the new clips, right? So you can still use that data for training on the clips, not the original data. So that's one way to go around it. Yeah.
Fantastic. So one of the questions here is the following. The same packages shown in the webinar can be used to analyze the whole community audio without identifying species?
No, well, it depends. But I try to be cautious about the context of the type of data you can use for. This is the home package that does this automatic detection. The methods are relatively simple. So, for instance, the energy-based detection requires good recordings, like good signal-to-noise ratio, and the template-based detection requires highly stereotyped signals, right? If you have something that is unlikely, that varies too much, so it's unlikely that you will pick up a single template that will look like any other sound that you are looking for, then that won't work. So you need to be aware of those, the context in which they are supposed to work, and mostly for a single species. Of course, you can get like a community analysis if you train different models for different species. But it's not something that is simplifying the model. The model is, or the functions are not aimed to do automatic detections of many templates or species at the time. Yeah.
Right. And I think there's a question here that's related to that. So for the auto call detection, are there ways that you deal with recordings in which there's a lot of overlapping sounds, even overlapping calls of the target species, like when you're recording a group of animals?
Yeah, well, I think that could be a problem. I haven't tried on that. So this package has been out for a year, and I tried that with lab recordings and also with passive acoustic monitoring of a single frog species. And in both cases, it worked well. Even for when you have some overlap of sounds from the same species. So you can get a good detection on that. But of course, if you have too much like signal jamming going on, and it's hard even for us to see the signal in the spectrogram, it's unlikely that it's going to be picked up by the model. Yeah, and I think there are some people working with machine learning, AI algorithms, to being able to, when you are looking for several species and they are kind of jamming or overlapping in time and frequency, then there are ways to tell them apart. But this like active like area of research, and I don't think those tools are out there that much. So far I think that's a big problem that is not easy to handle for bioacoustic analysis. Yeah.
Great. All right, so I think we are at the limit of our time slot. Marcelo, thank you very much for the presentation. I think it was really nice, and it's going to be a really great resource that people can come back to and look at our markdown, look back at the video. That was really great.